today's lab, we're going to look at taking DNA and how we use DNA to make proteins. We've talked a lot about proteins throughout the semester. Proteins pretty much control everything within a cell. There are checkpoint proteins that we saw are important in cell division, transport proteins that help get molecules in and out of a cell. We saw that proteins can act as enzymes or catalysts to speed up chemical reactions. Proteins are part of the structure of our tissues in our cells. So today we're going to look at how we make proteins. And the information for making proteins is held in our DNA. And remember, our DNA is within the nucleus of our cell. And we make proteins at the ribosomes, which are attached to rough ER or are found in the cytoplasm. So we have to get the instructions for making our proteins from the DNA in our nucleus to the ribosomes that are outside of our nucleus. We don't want to have to keep taking our DNA in and out of the nucleus in order to make a protein. So we're going to have a messenger that's going to carry that information, the directions for how to make a protein, and is going to carry that information from the nucleus to the ribosomes. And our messenger is going to be a type of RNA, one that we call mRNA, messenger RNA. Now if you think back to when we talked about our biomolecules, we said DNA is double-stranded, 2 strands of nucleotides bonded together. RNA is a single strand. So we're going to have to take that double-stranded DNA molecule, use it to make a single-stranded messenger RNA, and then be able to decipher the information in messenger RNA to make a protein. So here's a section of DNA, our nucleotides. And if you remember, in our nucleotide sequence, when we go DNA to DNA, A bonds to T and C bonds to G. So here's our section of DNA. In our match to that, our second strand, G would go to C, T would go to A would go to T, and so on. So there's our double-stranded DNA molecule. This bottom strand we are going to designate as our template strand. Our other strand of DNA we are going to designate as our complementary strand. You will always use the template strand to make your messenger RNA, and that is important. You will get a different nucleotide sequence if you use the template strand than you will if you use the complementary strand. We will always tell you which strand is the template strand. You need to make sure you read and know whether we're talking about complementary strand or template strand. So, RNA is single-stranded. We already know that difference between DNA. But RNA also doesn't have the nucleotide thymine. It has uracil. So now when we go from DNA to make RNA, A is going to bond to U, T still goes to A, C goes to G, and G goes to C. So here's our template strand of DNA. We're going to use that to make messenger RNA. And I'll do that in green. So C here is going to go to G. A is going to go to U. T is still going to go to A to U, G to C. This is now our strand of messenger RNA. We have now undergone transcription. We have taken DNA and used it to make messenger RNA. We have rewritten that information, but we're still in the language of nucleotides. DNA is nucleotides, messenger RNA is nucleotides. We now have to decipher the nucleotide language into the language of amino acids, how our proteins are made. Messenger RNA is going to leave the nucleus and it's going to go to the ribosomes, where proteins are made. Messenger RNA is written in 3-letter sequences. Every 3 letters is going to code for an amino acid. Those 3-letter sequences we refer to as a codon because it codes for an amino acid. So these 3 letters will code for an amino acid, these 3 letters will code for an amino acid, and these 3 letters will code for an amino acid. In a couple of minutes I'll show you, okay, that amino acid chart and how you can read your messenger RNA. But for right now, okay, this 3-letter codon is going to code for the amino acid VAL. 3 letters, 1 amino acid. Our next set of 3 letters in our messenger RNA is going to code for the amino acid SCR. In our last 3 letters in our sequence is going to code for SER again. So we've taken DNA, we've converted it through transcription into messenger RNA, and we've now translated messenger RNA into amino acids that make our proteins. Transcription is just rewriting our nucleotides from DNA into messenger RNA. Translation is going from messenger RNA to our amino acid sequence. So think about when you translate from one language to another. We are translating from nucleotide language to amino acid language. So how do we make proteins? We said proteins are made by bonding amino acids together. There are only 20 different amino acids that can be bonded in different orders to produce the thousands and thousands of proteins that organisms need. We have 4 different nucleotides in our messenger RNA. We have A, U, C, and G. And those nucleotides are going to code for our amino acids in groups of 3. Therefore, we have way more nucleotide combinations than we have amino acids that we need to code for. So there's some redundancy in our genetic code. And if we look here, there are 6 different ways to code for the amino acid leucine. Any one of these messenger RNA codons will code for the same amino acid. That's what we refer to as redundancy. That's important. If you have a mutation in your DNA, which is a change in the nucleotide sequence, it may not result in a different amino acid being put in place. That messenger RNA may still code for the same amino acid, therefore still produce the same protein. So let's look at our example that we had when we made our messenger RNA that had the first codon, GUA. The way the chart reads, the first letter in the codon goes along the left-hand side. The second letter in our codon sequence is across the top, and the third letter is down the side. So we had G, we're going to start down here. We had U, we're going to go to the first column, and then we had A. And GUA codes for valine, or abbreviated VAL. Our second codon was UCC. U's at the top, okay? C is in our second column, and we come down to our third codon of C. UCC codes for serine, or SER. Our third codon was AGC. So here's our A codon, here's our G codon, and we come down to C. And notice AGC also codes for the amino acid serine, or SER. So our sequence in our short little amino acid sequence we were putting together was VAL, SER, SER. Proteins will be made of thousands of amino acids that get bonded together. All proteins will begin with AUG, methionine. That's our start codon. There are 3 codons that code for stop. Once the stop codon gets into the ribosome, no more amino acids get attached in that sequence. So if a mutation arises and we get a stop codon put in before the end of that protein sequence, no more amino acids get added. And now we have a protein that's not going to be the right length, not going to be the right shape, and therefore will not function the way it's supposed to. All living organisms read this genetic code the same way. Humans, bacteria, plants, animals all read the genetic code the same way. That is why we can take a human gene for insulin and we can put it into bacteria. And the bacteria will read that gene the same way a human does and will produce human insulin. The genetic code is universal. And it's important because each codon will only code for one specific amino acid. However, we have multiple ways to code for that same amino acid. The genetic code is amazing. It's a way to store information and then be able to use that information to produce our proteins. This table is found in your textbook on page 182. So you can use that table as you work your way through the activities in the lab manual today. Activity 1. I'm going to work through activities 1, 2, and 3 to show you how transcription, translation will work in order to produce a very short protein sequence. So in your lab book it says to lay out our DNA nucleotides in the sequence TAC, GGC, CAT, and TTC. That's our DNA nucleotide sequence. That is also going to be our template sequence in order to make our messenger RNA. The first thing we're going to do with our DNA sequence is we're going to make our complementary strand of DNA. So remember, DNA to DNA, A goes to T and C goes to G. So our top strand in our complementary DNA is going to have the sequence ATG, CCG, GTA, and AAG. There is our double-stranded DNA molecule. Now we're going to use just the template strand to undergo transcription and produce our messenger RNA. Remember, DNA to RNA. A goes to U, T goes to A, C goes to G, and G goes to C. So our first 3 nucleotides in our DNA template are TAC. That will code for AUG in our messenger RNA. Our second group of 3 is GGC, and that corresponds to CCG in messenger RNA. Our third group, CAT, is going to code for GUA in messenger RNA. In our fourth group of codons, TTC is going to code for AAG. We have now made messenger RNA. We have undergone the process of transcription. Now we're going to undergo translation, and that is activity 2 in your lab manual. We are going to read our messenger RNA in groups of 3. Each group of 3 is a codon and codes for an amino acid sequence. So using our genetic code chart, we look up AUG, our first codon. And AUG codes for the amino acid MET, methionine, our start codon. We move to our second codon, which is CCG. CCG codes for the amino acid proline, or PRO. Our third codon is GUA, and GUA codes for valine, or VAL. And our last codon is AAG, and that will code for lysine, LYS. We have now translated our messenger RNA into our amino acid sequence, and we've made our protein. Now we're going to undergo activity 3. We are going to make a mutation in our DNA sequence of nucleotides. Our first 3 nucleotides, TAC, our next group, GGC, and our third group, CAT, all remain the same. Our point mutation is going to occur in our fourth group of 3 nucleotides. Our original nucleotide sequence had TTC here. We've now made a mistake. A point mutation. 1 nucleotide has been changed. Instead of having TTC, we have ATC. That point mutation now changes the nucleotide sequence in our complementary strand. And now we have in our complementary strand TAG as our DNA sequence instead of AAG. When we take that DNA template strand and we transcribe it into messenger RNA, our first 3 codons are going to be the same: AUG, CCG, and GUA. We made no changes to those nucleotide sequence in our DNA. It's our fourth codon that's going to change. Our fourth codon is now going to be UAG instead of AAG. So now when we take our messenger RNA and we translate it into our amino acid sequence, our first 3 amino acids should be the same. We did not change those codons. So again, we have MET, PRO, and VAL. Our fourth codon and our fourth amino acid are going to change. Our codon UAG now codes for a stop codon. We get no amino acid added after valine. Our original protein consisted of 4 amino acids: MET, PRO, VAL, and LYS. Our mutated protein now only has 3 amino acids: MET, PRO, VAL. That protein will not have the same shape that it was supposed to have. And when we change the shape of a protein, we change how that protein functions. This protein that we have just produced will not do the function it was supposed to have. It has been denatured because the nucleotide sequence in our DNA was altered. You will be responsible for completing activities 4 and 5 in your lab manual on your own. But I will give you directions and help as to how to complete those activities. Activity 4, you're going to work backwards. Your lab manual has given you the amino acid sequence. You need to use the genetic code chart and look up that amino acid on the chart and then what the corresponding messenger RNA codon would be. I'll do the first one for you. Methionine. You find methionine or our start amino acid here. The only choice you have for the codon is AUG, and you would write that in your lab manual. The next amino acid in your sequence is tryptophan. You would find tryptophan on your chart, and you would write down the messenger RNA nucleotide sequence, the codon for that amino acid. And you'd go on to your third and fourth one. Keep in mind, lysine and arginine have multiple options for a messenger RNA codon. You can pick any of those options and record in your lab manual. From there, you're going to decipher your template strand of DNA. When you do that, you're going to read this chart backwards. If your messenger RNA has a U in it, your DNA will have an A. If your messenger RNA has an A, DNA will have a T. Messenger RNA has a G, DNA has a C. And our last one, if messenger RNA has a C, your DNA would have a G. You will construct and write in your lab manual your template strand of DNA, and then use that template strand of DNA to produce your second or complementary strand of your DNA molecule. And that will work you through activity 4 in your lab manual. Activity 5 is a protein challenge for you to work out. In your lab manual, you are given a gene sequence. That is your DNA strand. Please read the directions to know if that is your template strand of DNA or your complementary strand of DNA. Use your template strand of DNA to make your messenger RNA sequence. Use your messenger RNA sequence to determine your amino acid sequence using the genetic code in your textbook. Remember, messenger RNA gets read in groups of 3. Each group of 3 nucleotide is a codon that codes for an amino acid. When you get your amino acid sequence in your lab manual, you will match it up to the protein ID key at the bottom. You will have 3 proteins to decipher. You have 4 options down here. Your amino acid sequence will match one of those sequences in our proteins. If they don't match, you've made a mistake. Go back and redo your messenger RNA sequence and make sure that your codons are correct. Those amino acid sequences should match up to one of these below. Okay, activities 6 and 7 are going to relate to what we're going to be doing later on in the lab, where we actually separate out DNA based on the sequence in the length of that DNA strand. So now we're going to introduce the concept of restriction enzymes. Restriction enzymes are like molecular scissors. They will cut a strand of DNA only if it finds a certain sequence that it's looking for. Restriction enzymes are derived from bacteria. Bacteria do not have immune systems. So when bacteria get invaded by viruses, they use restriction enzymes to cut up the DNA of that virus to destroy it. So the restriction enzyme we're going to be focused on is one called MST2. MST2 looks for a specific nucleotide sequence within the DNA molecule. It looks for CCT, N, followed by AGG. Well, N's not a nucleotide we've been talking about. N can be any nucleotide. It could be an A, a T, a C, or a G. That nucleotide isn't important. It's that CCT precedes that nucleotide and AGG follows it. And when that happens, the restriction enzyme MST2 will make a cut between the C and the T. Now remember that DNA is double-stranded. So we have two strands of DNA that we are going to cut. MST2 will read our top strand going from left to right. And it will read our bottom strand going from right to left. So here's our DNA molecule. CCT, G, AGG. And our bottom strand, read from right to left, is CCT, C, AGG. Now we're looking for this sequence, CCT, followed by any nucleotide, and then AGG. And when MST2 finds it, it makes a cut between the C and the T through just one strand of DNA. And now it reads this strand coming this way, CCT, followed by any nucleotide, and then AGG. And again, it's going to cut between the C and the T. And now we have an uneven cut. We've taken one piece of DNA and we have divided it or split it into two strands. Notice, however, it's uneven. This strand, the two C's, are bonded to the two G's, but our A, C, and T are not bonded to anything. That is what we call a sticky end. And notice our second strand that we've cut. Again, the C's are bonded to the G, but again here, we have no nucleotide bonded to it. Both of these areas are sticky ends. The sticky ends can go back together if they find a complementary nucleotide. But right now we've taken one DNA strand and we've cut it into two smaller strands. Now we're going to look at how that affects the hemoglobin gene, which is in your red blood cells. Here in your lab manual is the sequence that we're going to start with. AGG, CCT, GAG, GAG. That's our complementary strand of DNA. From there, we're going to make our template strand. Remember, DNA to DNA is A to T and C to G. So A bonds to T, G bonds to C. So remember, this is our DNA molecule. This section of our DNA molecule is part of the hemoglobin gene, part of the gene that produces the protein hemoglobin that helps to give our red blood cells their shape and to be able to carry oxygen. So from our template strand of DNA, we're going to make messenger RNA. So remember, DNA to RNA. T still gonna go to A, C still gonna go to G still gonna go to C, but remember, there is no thymine in messenger RNA. A will go to U. So when we make our messenger RNA, we have AGG, CCU, GAG, GAG. And this is our messenger RNA molecule. And again, we take that messenger RNA molecule and we read it in groups of 3. Each group of 3 is a codon. And that codon codes for an amino acid. So AGG is going to code for the amino acid ARG. CCU is going to code for the amino acid PRO. And GAG is going to code for GLU, and now we have GAG again. And now we have GLU again. So this section of our normal DNA that codes for hemoglobin would have the 4 amino acids ARG, PRO, GLU, and GLU in sequence. Now, what would the restriction enzyme MST2 do when it comes across this section of DNA? Well, here we have in our Legos, okay, our DNA model made up. This is corresponding to our complementary strand at the top. So we have AGG, CCT, followed by GAG and GAG. And here's our complementary strand at the bottom. TCC, GGA, CTC, CTC. So we're going to add our imaginary MST2 restriction enzyme to this DNA molecule. Remember, it reads the top strand from left to right and the bottom strand from right to left. And it is looking for that sequence, CCT, CCT, followed by any nucleotide, and then AGG. We have that sequence in the top strand, and it's going to make a cut between the C and the T. Now when we read our bottom strand, we have CCT, any nucleotide followed by AGG. And again, it's going to make a cut here. And we are going to end up with 2 pieces of DNA. So here's my AGG, here's my CCT, GAG, GAG, and we're going to make a cut right here. And now we're going to read the bottom strands. Okay, and again we have CCT, followed by any nucleotide, AGG. And we're going to also make a cut. MST2 has now cut that DNA molecule into 2 smaller strands. We went from 1 large DNA molecule to 2 smaller strands. We can separate DNA molecules by their size. And we'll do that in our lab next week. But now we're going to look at what happens when you have a mutation to your DNA molecule. Now we're going to show what happens when a person has sickle cell disease. Sickle cell disease is a point mutation, 1 change in a nucleotide sequence in our strand of DNA. And that changes the hemoglobin protein that's produced. The hemoglobin protein will have a different shape, and we'll show you how. So the 8th nucleotide in our strand, 1, 2, 3, 4, 5, 6, 7, 8, originally was GAG, but a mistake was made, and a T was put in instead of the A. So now we have GTG, GAG in our complementary strand. Our template strand then is TCC, GGA, CAC, CTC. This nucleotide bonding has been switched. We have a T here instead of an A. We have an A here instead of a T. What does that do to the messenger RNA molecule that we produce? Well, this is still going to be the same. We still have AGG. We still have CCU. But now we have GUG, GAG for our messenger RNA. And now when we take those messenger RNA codons, again in groups of 3, these two and this one won't change. We did not change the nucleotide sequence. So it will still code for ARG, PRO, and GLU. This one has changed. GUG now codes for the amino acid VAL. Inserting VAL instead of GLU changes the shape of that hemoglobin protein, changes the shape of our red blood cells. So a person with normal hemoglobin has red blood cells that look like this. A person with sickle cell disease will have red blood cells that look like a crescent moon. They don't live as long as normal red blood cells. They don't carry oxygen as well. They also will tend to clump and clot together. They don't flow as easily. And so that's going to result in a lot of medical problems for a person with sickle cell disease, all because of one nucleotide change. It also affects how MST2 will react to that DNA sequence. So remember, MST2 is looking for CCT followed by any nucleotide and then AGG. So what do we have in our sickle cell gene? We have CCT followed by any nucleotide, but now we have TGG. MST2 will not cut that DNA strand. It will be the template strand going the other way. So looking for CCT, we have CCA, followed by any nucleotide, AGG. It's looking for CCT. It will not cut that DNA strand. A person with sickle cell disease, their DNA stays as one long piece of DNA when MST2 has been added to it. Remember, our normal DNA got cut into 2 smaller pieces. So in next week's lab, we can separate out DNA based on its size. The longer piece of DNA will not move through our gel electrophoresis as fast or as far as our 2 smaller pieces. And that's the premise we'll use to be able to tell next week whether someone has the 2 normal genes for hemoglobin, whether someone has sickle cell disease in the 2 sickle cell genes, or whether they are a carrier for sickle cell disease, meaning they have 1 normal gene and 1 sickle cell gene.